Hi, my name is Hannah McChesney and I'm a member of the lab Bugs and Bugs, or microorganisms and bees and other insects. We study the microbiota that live in the guts of insects and what function they serve for the host. We combine entomology with microbiology and pretty much get the best of both worlds. In the case of honeybees and bumblebees, which are the most widely studied, the bacteria and host have a co-evolved mutualistic relationship in which the bacteria receive nutrients and the host obtains resistance to parasites and can better digest pollen. The honeybee has a core microbiome of 9 bacterial species, and the majority of this bacteria is found in the hindgut, the ileum, and the rectum. However, bacterial and host interactions in other bee and wasp species are highly understudied, and I study the microbiota of native bees and wasps in order to determine if there is a core microbiome in understudied native species, how the bacteria is likely conferred, and what patterns can be observed between related bee and wasp species. We go out into nature on sites we are okay to collect on and collect insects like so. Then we bring them to the lab and dissect them to remove the gut. We then perform phenylchloroform DNA extractions to extract the 16S bacterial DNA from the insect's gut. After this, we perform polymerase chain reaction to amplify our target sequence in the 16S gene. After PCR, we run gel electrophoresis to view whether or not any DNA was amplified. If so, we submit these samples for Sanger sequencing in order to receive back data that can tell us the identity of the bacteria. Once we know this, we try to determine if there are any patterns between species or evidence of a core microbiome. Very little is known about the microbiome of native pollinators, and so the results that we receive from this research will provide us with important and novel information. Native pollinators provide us with essential ecological and economical functions, and an understanding of the microbial contents of their guts and their function can be essential to ensuring their health and understanding the biological mechanisms at work.